This video covers Chapter 18 Notes, which is looking at gene expression regulation. We're going to start by talking about prokaryotes and how they regulate gene expression because it turns out it's a little simpler and a little easier to understand than with eukaryotes. So both prokaryotes and eukaryotes regulate gene expression in response to their environmental conditions. Gene expression regulates development and is responsible for differences in cell types because we know that all cells have the full set of DNA, but not all proteins are made and used in every cell. So because we have specialized cell, only certain genes will be on in that cell type. And all of this has happened because natural selection favored bacteria that only express the genes that they need. And so our example here and throughout when we're talking about bacteria is going to be E. coli. And this E. coli is, the strain that lives in our human digestive system. And one of the examples we'll look at is with tryptophan, which is an amino acid. So in E. coli, if the environment needs tryptophan, the cell makes it. But if there's enough tryptophan, the cell stops making it. And so with controlling these metabolic pathways, the cell will either adjust activity of the enzyme present or adjust the production of the level of certain enzymes. And bacteria do that through the operon model. And this is a picture diagram of the tryptophan operon and how that works. And we'll go through the explanation here that will help make this more clear to you. So operons are a cluster of functionally related genes that can be controlled by a single on-off switch. So the operon is going to control whether those genes are on or off. The switch is a DNA segment called an operator that's usually going to be found within the promoter region. And the operon is the DNA that includes the operator, promoter, and the genes. So the whole thing is the operon, and then the operator is the on-off switch. Operons are switched off by a protein repressor, and that repressor binds to an operator and blocks RNA polymerase, which in turn prevents transcription. The repressor is a product of a separate regulator gene that's going to be in a different location. And the repressor can be in an active or inactive form, depending on what other molecules are present. A co-repressor is a molecule that works with a repressor protein to switch the operon off. And so using tryptophan as our example, as I said, E. coli synthesizes tryptophan when it does not have enough. The default in E. coli is that the TRP, which is short for tryptophan, the default is that operon is on, meaning tryptophan is transcribed. And so as the amount of tryptophan builds up, it's going to bind to the TRP repressor protein, which turns the operon off. All right, so the presence of tryptophan will cause it to bind to the repressor, and that's going to turn the operon off so that tryptophan will no longer be made. And here is a picture example of that operon, as you can see there. There are repressible and inducible operons, which are two types of negative gene regulation. A repressible operon is usually on, and that would be like TRP. An inducible operon is usually off, and an inducer inactivates the repressor, which turns on transcription. And our example there that we'll look at with E. coli is the lac operon, which has the genes for breaking down lactose. So an inducer inactivates the repressor to turn the lac operon on. And here is an example or picture example of the lac operon. So when lactose is absent, the repressor is active and the operon is off. And when lactose is present, the repressor is inactive and the operon is on, because it's not going to waste its time making things that'll break down lactose if there's not going to be any around. Inducible enzymes usually function in catabolic pathways, and their synthesis is induced by a chemical signal. With repressible enzymes, they usually function in anabolic pathways, and the synthesis is repressed by high levels of the end product. Both TRP and lac operons involve negative control of genes because the operons are switched off by the active form of the repressor. Some operons are subject to positive control through a stimulatory protein. So E. coli prefers glucose over lactose. And so if both are around, it's going to use up the glucose first and then resort to breaking down lactose, which is made up of a glucose molecule and a galactose molecule. And so it's going to break down the lactose so it can get the glucose that it needs to do all of those wonderful cellular functions that use glucose. And the example here is the cyclic AMP receptor protein, which is an activator of transcription, and for short, we'll call it CRP. So when glucose is scarce, CRP is activated by binding with cyclic AMP. And then that activated CRP attaches to the promoter of the lac operon, which accelerates transcription. And then as glucose levels increase, the CRP detaches and transcription returns to the normal rate. So that would be an example of positive gene regulation, which you can see a picture of here. So with the lac operon, that has two different things that can kind of increase or decrease transcription. So next we're going to look at eukaryotic gene expression. And these genes are turned on or off in response to internal and external environmental signals. And as I said, the gene expression regulation is going to be more complex in eukaryotic organisms, which is why we look at prokaryotic organisms first. So here is the situation. Almost all cells have an identical genome, and differences in cell types are the result of differential gene expression. Any abnormalities in expression can lead to disease or cancer, and it's regulated at many states or stages, which is most often going to be transcription. And so there's a sum up of those processes here in figure 18.6. One way gene expression is regulated is through the regulation of chromatin structure. And genes within highly packed heterochromatin are not usually going to be expressed. Okay, so it's going to be wound so tight that those genes are just simply not accessible to RNA polymerase. And chemical modifications to histones and DNA of chromatin influence chromatin structure and therefore gene expression. Next, we'll look at histone modifications and DNA methylation. Histone acetylation is when acetyl groups are attached to an amino acid in a histone tail, which opens up the chromatin structure and promotes transcription. Methylation, which is simply the addition of methyl groups, condenses chromatin and reduces transcription, which can cause long-term inactivation of genes. So that chromatin is so condensed that that gene is probably not going to be transcribed at all. And you can see examples of both of those there in figure 18.7. Chromatin modifications may be passed on to future generations of cells. So as methylation happens, for example, and all those sort of things, as that cell divides, maybe that's going to be passed on. Epigenetic inheritance is the inheritance of traits transmitted by mechanisms that do not directly involve the nucleotide sequence. So that has nothing to do with the order of the nucleotides, but everything to do with how accessible the gene is. There can also be regulation of transcription initiation, and there are chromatin modifying enzymes that provide the initial control of gene expression, which will also make that region of DNA more or less accessible. And next, we'll look at the organization of a typical eukaryotic gene and its transcript, which you see in figure 18.8. 8. So we've got an enhancer upstream, and then we've got the promoter, and then our introns and exons, followed by the poly A signal sequence in the termination region, and then we get our primary mRNA or our pre-mRNA that is going to go through processing, and then we have our final mRNA transcript that will be able to go through translation. And there are control elements, which are the segments of non-coding DNA that serve as binding sites for transcription factors. And we talked about this some with protein synthesis, but we'll look at some more detail here. Transcription factors help regulate transcription, and these are critical for precise regulation of gene expression in different cell types. General transcription factors are essential for transcription, so without them, not going to happen. In eukaryotes, high levels of transcription depend on the control elements interacting with specific transcription factors, and those are especially important for genes that are not expressed all the time. General transcription factors are usually going to be around the promoter, and RNA polymerase requires transcription factors to initiate transcription. We already know that. Some of them are going to bind to the TATA box within the promoter, and some may bind to proteins like other transcription factors or RNA polymerase itself. And all of that has to be in place before transcription can start. So whether or not those are there is going to influence whether transcription happens or not. Enhancers, next we'll look at enhancers and some specific transcription factors. Proximal control elements are located near the promoter. Distal control elements may be far away from that gene or located in an intron. And groupings of those are called enhancers. And each enhancer is only going to be associated with one gene. Not many, just the one. An activator is a protein that binds to an enhancer and stimulates transcription. And it has two domains. One binds DNA, and the second activates transcription. The bound activators facilitate a sequence of protein-protein interactions that result in transcription of a given gene. All right. So it's going to bind to that enhancer to help stimulate transcription, which you can see what that is going to look like here in figure 18.10. There are a combination of control elements that activate transcription only when the appropriate activator proteins are present. And so there are many possible combinations that can lead to activation and therefore transcription, which you can see two different things that can lead to that in figure 18.11. There's also some coordinately controlled genes in eukaryotes. So co-expressed genes are not organized in operons. They can be scattered over different chromosomes, but have the same combination of control elements. The activator proteins in the nucleus recognize specific control elements and promote simultaneous transcription of those genes. Another regulation thing would be post-transcriptional regulation, which we looked at with protein synthesis, with RNA processing, and alternative RNA splicing, which you can see here in figure 18.13. That one primary transcript can give two different mRNAs. That'll lead to two different polypeptides. Another thing that controls gene expression is the initiation of translation and mRNA degradation. So translation of selected mRNAs can be blocked by regulator proteins that bind to sequences or structures of the mRNA. Translation of all mRNAs may be regulated simultaneously. An example of that is that translation initiation factors are simultaneously activated in an egg following fertilization. The other issue is that the lifespan of mRNA is not endless, and there will be a specific nucleotide sequence that will influence its lifespan. So it's not like you make the mRNA one time and you're good for life. Those mRNAs have to be continually made for continual protein synthesis. It can also be regulated with protein processing and degradation. Um, Polypeptides will undergo processing after translation and get some chemical modifications. And the length of time that protein remains functional will be regulated by selective degradation. And so those cells will mark the proteins of, into your lifespan, we're going to get rid of you. Hopefully, there's some more already made and ready to go. Next, we'll look at non-coding RNAs that do also play roles in controlling gene expression. For a long time, we used the term junk DNA to describe these are the regions that don't code for anything, but we're finding more and more often that they do something even if we didn't know about it. And so a small fraction of DNA actually codes for protein, and then a small fraction of non-protein coding DNA has the genes for RNA, like for making rRNA and tRNAs. And the rest of the stuff, we don't know what it does yet, but it wouldn't be there if it wasn't totally necessary. So it's helpful for one reason or another. And so first, we'll look at the effects of mRNAs by microRNAs and small interfering RNAs. MicroRNAs are small single-stranded RNA molecules that can bind to complementary sequences in the mRNA. So instead of it being single-stranded, that miRNA will bind to a single-stranded mRNA. And what that is going to do is block translation or cause that mRNA to be broken down. Small interfering RNAs or siRNAs are similar in function to miRNAs. They block gene expression, and we call those RNA interference. And those are used in the lab to disable genes to investigate their function. So a lot of what we know about what a gene does is scientists in the lab blocking that gene either from being transcribed or translated and seeing what happens. And that's how we have figured out which ones are super essential for living things. Chromatin remodeling and effects of transcription by ncRNAs will also help control gene expression. ncRNAs are non-coding RNAs, and some may bring about remodeling of chromatin structure, which we know makes genes more or less accessible for transcription. And in some yeast, siRNAs reform heterochromatin at centromeres after replication. And we also know that long non-coding RNAs or LNC-RNAs are a type of these that are responsible for X inactivation in females, where one of those X chromosomes becomes compacted and inactive. So it's the other one that is coding for all those genes. And there's a picture example here in figure 18.15 if you would like to read more about that. Next, we're going to look at cell programming and differentiation. So a fertilized egg gives rise to many different cell types during embryonic development, right? So the first several cell divisions, those cells have the ability to become any type of cell. If we're talking about humans, in the human body, or for whatever organism we're talking about, they can be literally anything. And at some point, they'll get to signal of you need to turn into this or that, and we get some specialization there. And so those cells are eventually organized into tissues, organs, organ systems, and then the whole organism. And gene expression orchestrates the developmental programs of animals. So first, we'll look at the genetic program for embryonic development. The transformation from zygote to adult it comes from cell division, cell differentiation, and morphogenesis. Cell differentiation is the process by which cells become specialized in structure and function. Morphogenesis is a physical process that gives an organism its shape. Differential gene expression from genes being regulated differently in each cell type. And the materials in the egg set up the program of gene regulation that is carried out as the cell divides. So based on how that gets divided, they'll get the signal to do different things. And to look more into that, we're going to look at cytoplasmic determinants and inductive signals. The egg cytoplasm has RNA, proteins, and other substances that are distributed unevenly across the egg. Cytoplasmic determinants are the maternal substances in the egg that influence early development. And as that zygote divides, the cells contain different cytoplasmic determinants, which lead to different gene expression further down the line. Another source of developmental information is the environment around the cell, so signals that it gets from nearby embryonic cells. Induction is when the signal molecules from the embryonic cells cause changes in nearby target cells. And so in figure 18.17, you can see the cytoplasmic determinants, again, unevenly scattered throughout, and also an example of induction by nearby cells, which is the classic signal transduction pathway that we all wish we didn't have to know and could forget about. Next, we'll look at sequential regulation of gene expression during cell differentiation. Determination is when the cell is committed to becoming a particular cell type, and that's going to happen before differentiation. So we have determination and then differentiation. Cell differentiation is marked by a production of tissue-specific proteins. So first it's determined of like, okay, I'm going to become this type of cell. And then once it starts making those specific proteins that are going to be found in that cell, then it is differentiated. And as our example here, we'll look at myoblasts. Myoblast cells are determined to form muscle cells and produce large amount of muscle-specific proteins. MyoD is a master regulator gene that encodes a transcription factor that commits the cell to becoming, specifically here, skeletal muscle. And so that's what we see here in figure 18.18 of what happens during that process. Another important part with cell differentiation is pattern formation and setting up the body plan. Pattern formation is the development of a spatial organization of tissues and organs. So in animals, that pattern formation begins with the establishment of the major body axes. Positional information is the molecular cues that control pattern formation, and it tells a cell its location relative to body axes and neighboring cells. And scientists have studied this extensively in fruit flies, which we just can't seem to get away from. But that's kind of where we figured out a lot of stuff about humans and with other organisms. Fruit flies are a monoorganism, so we study them and then figure out how that relates to us as humans. And Drosophila is our fruit fly genus that we love to learn about. And the cytoplasmic determinants in the unfertilized egg determine the axes before fertilization even happens. After fertilization, the embryo develops into a segmented larva with 3 larval stages. All right, so we can see that looking over here in figure 18.19, you can at the top see the full adult, the 3 body regions, and then you can see where that's coming from the developing egg, mature unfertilized egg, fertilized, segmented embryo, and then finally the larva. The larva forms a pupa, and then that pupa undergoes metamorphosis into an adult fly. So it's not a long process, but it has to go through more steps for sure. The homeotic genes are what control pattern formation in the late embryo, larva, and adult stages. I don't want to spend a lot of time talking about that here because I feel like I've already been talking for way too long. But you should read more about this in your book. I'm not expecting you to have all of this stuff, memorized or anything, but I do think it's interesting and want you to have some ideas about the important role that those genes play in development. Finally, we're going to look at what happens when gene expression goes awry. Gene regulation systems that go wrong during cancer are the same ones that are involved during embryonic development. And there are different types of genes associated with cancer. They can be caused by mutations to genes that regulate cell growth and division. So when cell growth and division is not highly regulated and things are, dividing too quickly, there's more likely to be an error. That's when we can get the development of cancer. And what we already know is that that can be caused by a spontaneous mutation that happens for seemingly no reason, or an environmental influence like from a chemical or UV radiation. Oncogenes are what we call cancer-causing genes, and proto-oncogenes are the corresponding normal genes. And proto-oncogenes can be converted to oncogenes by movement of DNA within the genome, amplification of a proto-oncogene, and point mutations. We have normal and the ones that generally can lead to cancer. And so there's a picture example of all of that here in figure 18.23. You can see our proto-oncogene and what can happen with that as it can either function normally or get turned into an oncogene. There are also tumor suppressor genes that normally inhibit cell divisions, and mutations that decrease the protein products of tumor repressor genes may contribute to cancer onset. Of course, we know cancer is more complex than one or two things. It's, lots of different things interacting together all at the same time. Tumor suppressor proteins are important because they can help repair damaged DNA, control cell adhesion, and act in cell signaling pathways that inhibit the cell cycle. Because again, we don't want those cells going through the cycle faster than they should. That's when we can have more room for error. So let's look at an example of interference with the normal cell signaling pathways. Mutations in the RAS proto-oncogene and p53 tumor suppressor gene are common in human cancers. Mutations in the RAS gene can lead to production of a hyperactive RAS protein and increased cell division. The RAS protein is a G protein that relays a signal from a growth factor receptor on the cell surface, and the response to the resulting cascade stimulates cell division, which we see here in figure 18.14. So we've got our growth factor and our receptor, and that G protein that's in our protein kinases, and eventually the response is a protein that stimulates the cell cycle. But with a mutation, we have overexpression of that protein and increased cell division. More cell division than needs to happen. Mutations in the p53 prevent suppression of the cell cycle, and if the DNA damage is irreparable, p53 can activate the apoptosis genes that will cause programmed cell death, which is, similar pathway. We do not want increased cell division. And finally, we'll take a brief look at the multi-step model of cancer development. Multiple mutations are generally needed for cancer, and so that's going to increase as we age and get older. The cancerous cell usually is characterized by at least one active oncogene and a mutation of several tumor suppressor genes. And so here in figure 18.26, we have an example of colon cancer. So first we have the loss of the tumor suppressor gene, and a small benign growth or a polyp will start to form. And then we have activation of the RAS oncogene, loss of a tumor suppressor gene. And so now we have a larger benign growth. And then we have the loss of tumor suppressor gene p53 and additional mutations that lead to a malignant tumor. And so all of that are some examples of things that help regulate gene expression. Most of them occur during transcription, but some can happen before or, to block translation as well. And when we lose regulation, cancer or disease can happen.